Yes, I titled our presentation today Explorations, because we are researchers and we don't have definitive answers on any of this, but explorations of motor and cognitive changes associated with Parkinson's disease. Again, just a very quick overview. I'm going to start off by telling you a bit about what we do, but also tell you a little bit about what we don't do, to make that distinction clear. And then today's team, we have Elizabeth Cisneros and Sabrina Abraham, both members of the lab who are doing research, and much of that research involves working with people with Parkinson's disease. So they're going to tell you about some of the ongoing studies, and briefly we'll also tell you at the end a little bit about how you can get involved, meaning how, if you're interested in participating in our research programs, how to sign up. Okay, so we call ourselves, we spent lots of time trying to come up with a clever acronym, and we came up with COGNIAC, the Cognition and Action Lab, and so we celebrate, when people get their PhD, we have a little COGNIAC party and all. But it's to capture, as we said, we study human performance, and performance is not only about how we act in coordinated manners, but also how we think in coordinated manners. This building houses psychology, public health, and education. There's obviously some overlap between the three groups. Right across the street is the Brain Imaging Center, where we do some research, sometimes where we're doing brain scans, either structural or what we call functional. I wanted to highlight this because this is actually something, ribbon cutting party from 1 week ago, is that Berkeley's always had a lot of neuroscience research going on. Even though we don't have med school, we have a lot of people studying different types of diseases, including neurological diseases. They are scattered across different departments on campus. Some are in molecular and cell biology, some in public health, some in computer science, and so forth. They've just created a department of neuroscience on this campus. There aren't that many actually departments of neuroscience in the country, so this would be kind of a focused effort. Some of us are going to be jointly members of different departments. I remain in psychology and a member of the neuroscience, but it's going to be a way to help bring together a lot of the neuroscience research that goes on campus, including that there's quite a few different labs that are studying, maybe not Parkinson's disease directly, but some of the cellular molecular changes that accompany Parkinson's disease. So it's going to be great to have this new neuroscience department. It doesn't have one building right now. It's kind of an amalgamation of different buildings on this corner of campus, but I'm sure you'll be hearing things about research coming out of the Department of Neuroscience. We are one lab within the neuroscience psychology programs here. This is members of the lab. A lab at Berkeley usually is composed of some postdoctoral fellows. Sabrina is a postdoctoral fellow visiting us from Canada, and then graduate students, PhD students. Elizabeth's in the PhD program here. We like to, go on inspiring hikes to talk about ideas and just refresh ourselves here. So what do? We do a lot of what I call neuropsychological research, okay? I'm not an MD, so I usually cringe a little bit if people introduce me as Dr. Ivry, because that always has the wrong connotation there. And then you say you're a psychologist, and then of course they think, oh, you're going to do therapy. I know nothing about clinical psychology. No one in the lab really does. But we still are neuropsychologists, which means we're interested in the relationship of the brain and behavior. And it's sort of this triangle that's captured here. As psychologists, we're interested in how the mind works, how the mind allows us to create memories, produce skilled movements, solve problems, make decisions, and so forth. Of course, the mind is supported by the brain, but ultimately we also want to observe behavior. So it's that three-way, that triangle there, which is what we call cognitive neuroscience, capturing the idea that we're interested in the neuroscience, the biological mechanisms that support cognition, okay, support our thinking abilities, and so we have that name, the biology of the mind. Now, in our particular lab, these are the kind of key questions that we like to ask. How do we produce and control movements? I've long been interested in, I actually went to graduate school and I wanted to understand why some people were more skilled at doing certain actions than I was, for instance. I loved to play sports, but I wasn't very good at it, okay? So I had an interest in that. And that, of course, then leads to an interest not just about how do we produce the movements, but how do we acquire new movement skills. But then you quickly understand that actually what explains a great athlete compared to a not so good athlete isn't really anything to do, very little to do with the muscles or the body shape or the physique. There's probably some contribution there, but much of it is really about mental, about how we represent those actions, how we can quickly recognize what's the correct movement to make and so forth. So there's a very intimate relationship between skilled movement depends on skilled thought. And so that's become a very central focus there. And in particular, we've been interested in what is the role of the basal ganglia, and I'll tell you more about that, and you probably know some about this, because that's a key structure that's involved or affected in Parkinson's disease. So we're interested in the role of the basal ganglia and the cerebellum in supporting all of this. And I'll talk more about that in detail. And of course then, the neuropsychological part comes in about where I'm interested in how are these processes, this ability to have skilled thought and skilled movement, impacted by different degenerative disorders that's either affecting the basal ganglia, namely Parkinson's disease, or affecting the cerebellum, a condition that results in what's called ataxia. Now, we use many different methods in our research. We certainly study behavior, and if any of you participate in our experiments, you might have a sense of that, where we're asking you to either produce movements under certain types of conditions, or maybe looking at other things. Sabrina will talk about that later on. We also use some measures to look at the physiology while people are engaged in these behaviors. For instance, EEG is a way to look at, monitor brain activity by recording with electrodes placed on the surface. We do brain imaging right across the street here, where you can actually see how oxygen is being used by the brain while you're, say, doing a challenging task. And when you're doing a challenging task, as different parts of the brain become active, they demand a lot of oxygen, so we can monitor that. And we actually do some work on brain stimulation. Now, that sounds scary, but it's non-invasive, safe brain stimulation as a way to transiently alter a person's brain function and make inferences about how by altering the brain function do we actually observe changes in their behavior. But today we're really going to focus on the behavioral end of things, okay? Much of what we do is actually have people come in, participate in the experiments that are designed to test different hypotheses, and that's what we're going to walk our way through. But as a background, I wanted to give you a picture here of, this is the brain we typically think about, the cerebral cortex, the big giant thing that is really unique about humans. This is just highlighting in color the parts of the brain that seem to really be part of what we might call the motor system, that play an intimate role in the control, the selection, the production, the monitoring of movement. Now, I have a bit of a bias because I like to study movement, so I've given about half the brain over to the motor systems. If someone else was studying perception, they might steal some of these brain regions. But that, of course, is because, as people like to say, it doesn't do you any good to have a big brain unless that big brain's going to help you interact in the world. So everything's really about how we're going to be able to learn to take in information from the environment and produce skilled actions. This is what we typically think about. Here's the primary motor cortex that actually makes the muscles become active. What's missing in this picture, though, is what we call the subcortex. And the subcortex, as the name implies, is the parts of the brain that lie underneath the cerebral cortex. And it includes a number of different structures. The two prominent ones I've highlighted here are the basal ganglia, a collection of 5 different, what we call nuclei. Nuclei just means a cluster of different neurons that have some sort of commonality in terms of their function. And the cerebellum, this prominent structure down here. And one thing I want to point out is that these are 2 very ancient parts of the brain. All critters have a cerebellum and a basal ganglia, but many of them don't have any cerebral cortex. So these are, say, take something like a frog. It's capable of very complex interactions in the world. Okay, it might not have the flexibility that you and I have, but nonetheless, all of its sort of actions and thoughts and behavior perception are being controlled by the subcortex and very much relying on the function of the basal ganglia and the cerebellum, these 2 prominent subcortical structures that are intimately part of the motor system. And again, the 2 groups of patient populations that we typically work with are groups that have some sort of degenerative disorder of either the basal ganglia and the cerebellum. This is showing an MRI scan. So this is a person who's just lying in the scanner. We can take an image of their brain to see the different structures. And this is just showing what a brain would look like in someone who's neurologically healthy, showing again this sort of intact cerebral cortex. Here's the brain stem, the spinal cord going down, and here's that prominent cerebellum. This is beautiful, what they say, foliated structure. And there's certain degenerative disorders that target the cerebellum, okay? And that's what's shown here. You can see that the cerebellum has really shrunk a bit, right? You have these black spaces here, and that just means that there's been some loss of cell tissue. When you lose the cell tissue, it's replaced by the fluid that protects the brain. So the dark regions here are that extra fluid that's filling up where the cell death has occurred. And again, the condition that usually occurs with that is what's called ataxia. And people can make movements, but they're very staggered, they're very erratic. Okay, they might really feel unstable, so they tend to walk with a really wide stance. And when they reach for something, they kind of have a wobble to them. Okay? I don't have an MRI scan that really visualizes the changes that occur in Parkinson's. That's actually quite hard to see because it's a much more subtle change. But this is a slice, a postmortem slice, highlighting one part of the basal ganglia called the substantia nigra. Again, this might be familiar to many of you. It's notable because when you stain it or when you section it, you actually see that it looks much darker. That's because the cells in the substantia nigra contain iron, and iron is just a very black substance. This is, again, postmortem comparison here. This is a slice highlighting that substantia nigra with those black bands. And in Parkinson's disease, that's missing, okay? And that's because those cells in the substantia nigra have atrophied. They've died off. And so basically you're visualizing the absence of those cells in Parkinson's. And again, I heard this before, that all of us are showing a steady decline of the cells in the substantia nigra. Some people, it's a very slow decline, and others can be a rapid decline. And again, at some point when you've lost enough of those cells, that's typically when people start to develop the symptoms of Parkinson's. Lots of discussion, about, what are the causes of that? Does environmental agents accelerate this? Is there a genetic component, etc.? I'm not going to really talk much about that today. That's more on the clinical side of things. Now, again, as I said, why do we call this thing the basal ganglia when the different structures are actually located at very different places across the brain? These are the four prominent nuclei or clusters of neurons that form the basal ganglia. It's because it's kind of like a closed loop system. The input comes into the basal ganglia in the striatum, and it goes out through part of the globus pallidus with some internal processing. So it's kind of like a closed loop system. And I'll actually show you the diagram. And this diagram always frightens, especially the undergraduates in my class. You don't need to pay attention to most of it. I really just want to highlight again, at the top is the cerebral cortex, right? That big part of the brain. It's sending a giant input into the striatum, the receiving part of the basal ganglia. And this is really the main pathway we want to focus on, this direct pathway. The striatum is going to send a projection into the globus pallidus, and then it comes back up to the thalamus, and then relayed back to the cortex. So that's what I mean that there's a loop. There's an input all coming in basically to the striatum. A bunch of things are happening within here, and then the output goes back to where it came in. Okay, so the basal ganglia is in a position to modulate what's happening in the cerebral cortex. And the question is, what is it modulating? How is it changing the operation of the system there? Now here's the substantia nigra. That's the one that contains those dopamine cells. That's the part where we see the cell death in Parkinson's disease. So we're going to then ask questions about what is this, when you don't have this input here, how is that changing the whole physiology of the system? So that's the basic anatomy, but here's really the physiology we want to think about. First thing to really notice is that the output from the basal ganglia, that's going to go back up to the cortex, that's in red. And if it's in red, it means an inhibitory connection. So when this is active, it's basically shutting down the cerebral cortex, okay? So in a sense, what's the basal ganglia doing? Its main output is inhibitory. So when the basal ganglia is initially sitting there firing away when you're not doing anything, it's actually inhibiting the cerebral cortex, Okay, so keep that fact in mind. But when the cortex sends an input signal here, it activates this pathway. Now this is where it gets complicated. This is inhibitory also. So now we have inhibition of inhibition, or what we call disinhibition. So when the cortex sends a particular signal into the basal ganglia, the net result is we reduce this inhibition. We release the cerebral cortex. In a sense then, if the cortex sends an input, it actually allows the cortex to express its output, and the result is movement. So I think this next slide, I just double inhibition was just to make the point that this arrangement, this physiology here, is actually very unique. It's very few places in the brain do you see this double chain of inhibition. So it's something special about the way the basal ganglia operates. So rather than thinking about those details, here's kind of the metaphor I like to use to think about how the basal ganglia is contributing to movement. The cerebral cortex I envision as a bunch of little rivers, and each of those little rivers you can think about are all the possible things you could do at any point in time, right? I can talk to you, I can decide to pick up this marker, I can decide to kind of go out there and grab myself a donut or something. These are all the possible actions. At any one point in time, all those possible actions, maybe the context is telling me all these different, trying to like get me to do all these different things, like I smell the coffee and so forth. These are the possibilities, but the basal ganglia is sitting there saying, hey, don't take any action. You don't quite know what you want to do yet. So it's keeping the whole system in check. Now at some point though, I get a big enough activation of one of those possible responses and it breaks through the dam. That's that cortical input that gets strong enough. Yeah, I'm overwhelmed by that sense of coffee. I'm gonna dash out there and grab a cup. That's going to break through this dam, and that's going to be the way we can think about how a movement occurs, okay? So in a sense, the basal ganglia is allowing the cortex to figure out what's the right action to take at this point in time without that action being initiated. It's not until we get a very strong response here that we basically break through this inhibition and can initiate that action. And I call this, it sounds insulting, but I actually think it's a good thing in neuroscience. I call it a dumb selection mechanism. Okay, what do I mean by dumb? It means that the basal ganglia, if it's this dam, it doesn't need to be evaluating, is this a good action, is this a good action? It doesn't have to make the comparisons. All it has to say is, when something gets strongly activated, now I'm going to release it. Now I'm going to allow that movement to occur. So the basal ganglia's job is just to be that gatekeeper, saying, is something strongly activated? Then let that happen. Otherwise, I keep the whole system in check. That's kind of a nice feature because let's pretend, again, I'm deciding whether to pick up my computer bag or this pencil there. Well, as I think about those two actions, I'm activating the motor system that's going to be required to either make this action or this action. If as I thought about those things, my body would be like a little puppet, kind of playing out all the possibilities while I make that decision. But the basal ganglia, in a sense, says, don't allow any movement until one of those actions has really been strongly activated. So that's sort of the metaphor that we like to think about in terms of how the basal ganglia contributes to action. So let's then think about the key features of Parkinson's disease. And again, this was the basic picture I showed you before. The real change that occurs in Parkinson's disease is that because of the cell death down here in those dopamine-producing cells, we lose this excitatory input here. Well, if we don't excite the striatum, then we have a weaker output from the striatum. So we're weakening this inhibition. That's why this line is thinner than this line here. If this inhibition is weaker, now this inhibition becomes stronger because of that pairwise connection there. And so in Parkinson's disease, basically you have excessive inhibition of the cerebral cortex. And basically that means it's going to be a lot harder to initiate a movement, to break through the dam. And that's what I try to capture in this slide here. Imagine that this is the activation of the idea that I'm going to pick up this pencil, this marker here. And that's what this red line is signifying. Over time, at first I catch a glimpse of the marker. Then over time, the idea that this is a good thing to do builds up. That's what this red line indicates. Well, if I have a normal dam setting, normal level of inhibition, I don't need much input to break through the dam. If I call this the threshold is when the dam opens up. But if I have Parkinson's disease, because of that excess inhibition, I need a much stronger input signal from the cerebral cortex to actually break through the dam and initiate movement. And again, these are probably familiar to you, but I'll just show you a few videotapes to capture the idea. Oh, okay. This is a gentleman, again, with pretty advanced Parkinson's, has a lot of trouble walking. He doesn't need the cane to support himself. He uses the cane as a way to boost up that signal to get himself to put his legs forward there. That crazy sound is wife washing dishes in the background. But again, you really see some of the classic features of Parkinson's about, really it's that starting the movement. Once he gets going, you know, it might be with the small steps and so forth, but once he breaks through the dam, then he can get moving. But every time, you see it really clearly in this individual. Okay, he's walking there. Now he wants to turn around, and that's where you freeze. And it's sort of like he's trying to make a transition from walking in one direction to walking in the other direction. You got to break through that dam again, and that's where you really see the problem there. That's transitions from one motor state to another motor state is what's really challenging. You'll see it again with this gentleman. Gets going again, he's doing fine, but gets to the doorway, and it's sort of like, again, transitioning to a new state, he freezes up each time. Okay, and again, when you think about it this way, I'm saying that you need a stronger signal to actually break through the dam. There's all sorts of demonstrations out there about people maybe with advanced Parkinson's who have a lot of trouble lifting up their arm, but then they throw a ball at them and they go right up. Well, that's kind of an example about a very strong cortical input. That ball coming at you is sufficient to get this activation, give a boost to the external input, which will then allow you to trigger that movement by reaching that elevated threshold. Well, just briefly, I want to say how we study this in the lab. So this is the kind of motor task we use here, and I'll just walk you through that. Basically, people are going to learn to produce two different finger sequences, so they're fairly simple movements. In one case, you go index, ring, middle. So index, ring, middle. And we're going to play a letter on the computer screen. If we show you an A, that's what you do. You go 1, 2, 3. In contrast, if we show you a B on the screen, now you go index, ring. Oh, I did it backwards. Right. Index, I go index, middle, ring. Okay? So I have two different sequences. One is boom, 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 and the other is boom, boom, boom. One's associated with the letter A, one with the B. So what a person will do is they'll have to do two of these sequences in succession. We tell them go as fast as you can. In one case, you do the same thing twice, I, R, M, index, ring, middle, index, ring, middle. In the other case, they do index, middle, ring, and then index, ring, middle. Now again, we're trying to have good experimental control. The critical thing is what happens. In this case, you don't have to make a transition. You just do the same thing over and over again. It's like you've started walking, can I keep walking? In the other case, though, I have to switch. I have to make a transition. But we've set it up that you're actually making the exact same movement here. So we're asking how fast can you make that movement as a function of whether you're doing the same thing over again or making a transition. And that's what's plotted here. How quickly, measured in milliseconds, can you initiate that movement? So it takes about a half second. Now, of course, people with Parkinson's are slower than the control participants. We always expect to see that. But notice that there's very little increase in the control participants when they have to make that shift from doing the same sequence twice in a row to having to make the transition. But that's where you really see this larger cost, much more challenging for the people with Parkinson's, is when they have to make the exact same movement, but that movement is part of a transition, a shift, compared to when it wasn't part of a shift. So that's kind of the way we test this sort of idea, or develop the idea about this gating process, about basal ganglia being critical when you're transitioning from one state to another in these individuals. Now, what about cognition or maybe emotion and things like that? how might they be affected in the same sort of way? Well, if you look at the basal ganglia and you look at the anatomy, right, the big focus is on this loop right here. The motor cortex sends input to the basal ganglia. The basal ganglia sends output back to the motor cortex. That might be that gating operation. Here's all the possible movements. Select which one should be gated. But you actually see, in terms of the anatomy, that the basal ganglia is receiving inputs from all sorts of different parts of the brain. And there's the same sort of anatomical and physiological relationship at each of those places. And so that suggests that this gating operation may not just be relevant for motor control, but might also be relevant, say, for prefrontal, which is sort of more higher level thinking, or maybe emotion, associated with the limbic system. And just to give you a few examples then, so remember I showed you this picture here where the motor cortex might be the competition between all the movements I could make at any one point in time, the basal ganglia helping select or initiate one of those. Well, in the cognitive domain, I might be having all sorts of possible goals at any one point in time. Do I engage in a conversation? Do I cook dinner? Whatever it is, they're sort of competing with one another. And in the same way, maybe the basal ganglia is allowing us to choose which action, what do we focus on at this particular point in time. And we study that in a very similar way. It's actually an even simpler experiment. You're going to see a stimulus on the screen with a cue telling you whether to respond to the color or the shape. You have two keys. One key is associated with if it's red and you're supposed to attend to the color, that's the key you press, or green. And we do the same sort of experiment. We have a no-shift trial where on 2 consecutive trials you're responding based on the shape. But in the shift condition, you're now going to respond to the shape after having recently responded to the color. So again, here there's no shift, same mental state. Here I have to change from one mental state to the other, tending to the color or tending to the shape. And we get that same sort of pattern. Parkinson individuals are slower than the control participants, but there's an especially large increase in that added cost to shift when you're in that shifting condition. So this is again saying, same sort of general idea as a way to tie together some of the cognitive changes that are associated with Parkinson's disease, what's sometimes referred to as bradyphrenia or slowing of thought. People with Parkinson's frequently describe that they feel like they're just sort of stuck or in a fog is the way some of the people have described it to me. A way to think about how the same mechanism might produce a problem on the motor end and on the cognitive end. More recently, we've actually looked at very abstract mental shifting, okay? So you can all participate in this. I'm going to show you an equation, okay? Simple equation, not complicated mathematics. As quick as you can, everyone shout out true or false, okay? Ready to go? All right, here's your first one. 1, 2, 3. True. True, okay. I told you it wasn't going to be too tough. All right, next one. False. Okay. Let's go faster then. Don't wait to hear the rest of the crowd, okay? True. There you go. We got a spirited one there. True. Okay. True. Okay, we got a little mixture there. True. False. False. Okay. Perfect. You gave me great data there. I wish I'd recorded it. We have recorded it, okay? As you saw, right, we had some 2-number problems, some 3-number problems, some very easy, some more complicated, and so forth. All right? Here was the idea and the result. This is what the results. The results here, all I'm showing, the Parkinson patients were slower than the controls, as we always see. But the real question is, what's the added reaction time? What's the added time to respond when you go from doing a 2-digit problem to a 3-digit problem? And the idea was that, again, here I add 8 plus 3, get my answer. Here I do 4 plus 3, but now I have to mentally shift and add another number. And that's what's shown here. This is, it takes longer for the control participants to add 3 numbers compared to 2 numbers, but again, we see this added cost in the individuals with Parkinson. So just giving you an example about how that shifting idea may not only be relevant for the motor system, these slow transitions not only between different mental movement states, but all between movement states, but I've given you a few examples about how we study to look at this in terms of transitions between different mental states. We haven't done any work on here about emotional states, but it might be a way to think about the kind of mask-like behavior we think about in Parkinson's disease. To change from one emotional state to another is associated with certain types of facial expressions, and that may be part of the kind of difficulty that people with Parkinson's disease have in conveying different emotions. Again, this business that they're stuck in one particular state and can't transition to others. All right. Last thing I'll just say is about, if we think about the different interventions that have been used in Parkinson's disease, I like to think about it about, we've got the stuck gait. What are the interventions that people have tried to fix that stuck gait? Well, the medication, of course, and I imagine most of you are taking some sort of L-dopa medication. This pathway's gone, right? So we have reduced inhibition here, excessive inhibition of the cortex. With L-dopa medication, we're putting that pathway back in, presumably as a way to lower that tonic inhibition, to unfreeze the person, okay, to allow those transitions to occur. Not practiced all that often, but occasionally they still do what they call palidotomies, where they actually cut some of this tissue out in a palidotomy, brain surgery, that they remove some of this tissue. Why is it beneficial? Well, presumably because it's, again, reducing that inhibition of the cortex, so allowing the gait to be less stuck. DBS is more of a mystery, because with DBS we typically target this region over here. Notice that excites this, so if this excited it, you might expect that that would increase the inhibition. So it's actually counterintuitive in some sense, the benefits of deep brain stimulation, and we know it's amazingly effective. And it seems to be that when they stimulate this structure, they stimulate it at very high frequencies, very high levels of stimulation. And the consequence of that is it seems to actually shut this pathway off. That's one hypothesis. So by overactivating the structure, it becomes rendered functionally irrelevant. And again, if we reduce this excitation, we reduce this inhibition. So those are the kind of ways people are successful treatments we have, and how to think about them in the context of this gating. Anyhow, I'll be happy to answer your questions. Just briefly, some of you, how many of you participated in any of our studies? None. We got a bunch of one. John there, okay. If this, again, but I like to really emphasize that they're pretty simple experiments, but you've seen what they kind of look like. They're always sort of designed to be a one-off thing, meaning if you decide to sign up, you're going to probably be involved in a 1 to 2 hour experiment. You'll have an opportunity to come back and do other ones, but you're only really making a commitment for one particular session there. I will tell you, people tend to enjoy. We have some folks who've been coming back for 30 years with ataxia. Really like to emphasize though, again, there's no clinical benefit to our stuff. This is basic research. We are not clinicians. We're not involved in the treatment there. We do pay you for your time. Again, lots of people say, don't want to be paid. It's just to, again, really make clear that you are doing a service for us. We are not providing a service to you. It's supported by your tax dollars from the National Institute of Health. And so again, it's maybe more the intrinsic benefit about hopefully advancing our understanding about both how the motor systems work and how they are affected in people with different neurological conditions. So I'm happy to take any questions. Yeah.
When you mentioned the one role had an inhibitory effect on the inhibition, I can only think of first and second derivatives. And does anybody actually write on differential equations?
Oh yes, there are the most. Oh yeah. So the question is about, I'm showing this sort of these physiological things as one inhibitory signal affecting another inhibitory signal. And David's saying, right away, that's making him think about, is this mathematically amenable? Can I write a computer program